Now it's time for the first talk in the afternoon session. My name is Christopher Clemmensen, and I'm an associate professor and group leader at CBMR. And it's my pleasure to introduce the next speaker. So we have Dr. James Trevaskis from Eli Lilly, where he works within the obesity area, but within the broader area of cardiometabolic disease space. And I've followed Jim's work closely, actually, since I started working in this area myself. So actually, Jim was one of the first people that started studying the combination of different peptides and actually made, I think in 2014 or 13, the first Amylin GLP-1 fusion molecule. So way before we started to combine them and think about it as we do today, actually, there's already some publications. And another pioneering study by Jim is this observation that amylin actually seems to sensitize endogenous and exogenous leptin action, which is really exciting work, also done back already in 2008, actually in clinical, having already clinical data. And then he moved into industry, working in a company called Amylin Pharmaceuticals until that was kind of engulfed. And then more recently worked with a company called Gilead, focusing on fibrosis and fatty liver disease. And then more recently joined Eli Lilly and is now back in obesity. So we're very happy to have you here, and I'm excited to hear your talk. So please, the floor is yours.
Thanks, Christopher. Okay, thank you, Christopher, for the introduction. Thanks for the opportunity to talk and the invitation, Julene and Kay, and it's really great to be here and such a great crowd. First talk after lunch, I won't be upset if you close your eyes. Generally, I won't even see. But yeah, talking about obesity today, I think it's obvious Lilly's relationship with obesity therapeutics. These are my disclosures, which again, pretty obvious. So today, obviously, I'll focus a lot on GLP-1s, and then a little bit around future state, which probably not all GLP-1s, but it's very clear that GLP-1 receptor agonists are the most efficacious anti-obesity therapeutics on the market today. They deliver excellent greater than 20% weight loss pretty consistently, outstanding glucose and HbA1c control, and have really, innovated in this space. It hasn't happened overnight for those working in the space. It's progressively improved. There's enormous potential and interest in diseases outside obesity and diabetes for GLP-1s and other incretins, in particular cardiovascular disease, kidney diseases, liver diseases, even substance use disorders and disorders of dependence and reward, also being explored, neurodegeneration and Alzheimer's disease examples. Currently, they're a peptidic class of molecules, and their delivery is via injection. And I'll talk a little bit about innovation in that space to get to oral versions, which we're probably all aware of. And again, the response from all patients, while is very good on the whole, and as a mean, there is variability in patient response to incretins, as there is with almost every therapy. So I'll talk a little bit about some of what's coming through on the future side. I'm a huge believer in that we have to diversify the biologies, right? So discovery of novel biologies and targets to address unmet needs in the obesity and metabolic disease space. Well, I'm obviously a big advocate for, and hopefully you are too. So as I'm sure I don't need to tell this audience, glucagon-like peptide 1 and its derivatives and analogs thereof are based on an endogenous hormone, and it has pleiotropic effects in multiple metabolic and other tissues. I won't need to go into it a lot here. But the evolution of the GLP-1 class has taken a long time, right? And I think I would expect that there are probably faculty here that give classes on the evolution of GLP-1 and its early physiological evaluations. And over time, it really developed into a clinically relevant and analog, right, and compounds. The first GLP-1 receptor agonist, actually this year, 20 years ago, was exenatide. That's a very short-acting, very potent GLP-1 receptor agonist derived from the Gila monster. That's a little animal down in the bottom left there. It was thought to be a venom or a toxin from that model. It's actually not. It was just a peptide in the salivary gland extract. But that became the first GLP-1 receptor agonist by Amylin Pharmaceuticals out of San Diego. So I joined that organization about a year or so after it was approved and launched. It was a very exciting time in the space, and it was really nice to go and work with molecules that have such profound effects in rodent models as well as in people. But it wasn't a great drug. Obviously, it was twice a day injectable because it was so short acting, and that company, Amylin, was working on its long-acting version, which is the only one that uses a microsphere formulation for slow release, and that's by Durion, which was approved a few years after that. In between, Novo Nordisk here developed liraglutide, outstanding molecule, improving the PK instability to once a day. Eli Lilly evolved dulaglutide, which is an FC-based long-acting molecule for diabetes. It doesn't have much weight loss. And more recently, of course, semaglutide and tirzepatide have been launched. And these are really the two big molecules that dominate most of the discussion and conversation. In red, I've highlighted some of the new opportunities to come through in the multifunctional, bifunctional incretin class. These, of course, are more molecules with GIP receptor agonism or antagonism, as the case may be. It's a whole different conversation. More GIP, GLP-1 receptor agonists, glucagon-containing molecules, triagonists, and others. And so I think there's a lot of innovation in this peptide and biologic space. And of course, here this is easy to show the successes, but there's not a list here of the failures. Right? Not everything works in pharma. There's other companies have tried GLP-1s that have failed for some reason or another. Others have fallen away as out of favor. It's a really incremental. Leaders in pharma often don't like incremental advances, but I think, it's really hard. It's not an easy game to play to get a drug to market, and many factors, many unforeseen factors, prevent development of agonists and analogs along the way. So while it's a busy class at the moment and with two clear leaders in the space, there'll be more innovation, but others, do tend to drop off over time. So this is a one-slide summary of obesity trials, the therapeutic landscape. Phase 1's on the outside, going into phase 3's in the middle. I have one slide on each of these, and we'll just run through them over the next hour. No, I'm just kidding. We won't do that. But the take-home message is it's busy. This did not look like this 5 years ago. Okay, this is a rapidly evolving, competitive therapeutic space. And a lot of companies recognize that it's an opportunity from the commercial side, which hasn't always been the case. Historically, people ask, why didn't Byetta go for weight loss and obesity indication or pramlintide? And the answer is there was big commercial headwinds. No one wanted to stand up and say that the insurers will pay for it and there's a commercial case to be made. It became really not a biology issue or a pharmacology issue. It was just a commercial issue. So imagine transplanting this pipeline back 15 years ago and where we'd be today if some leaders and companies had really pushed ahead against those headwinds. So I'm not going to spend a lot of time talking about tirzepatide or semaglutide. They're out there. We've all seen that data. I think my topic today is really the future. And so I'll start with retatrutide. So this is the triagonist. This is the GIP, GLP-1, and glucagon receptor agonist that Lilly are developing. It's in late-stage clinical trials. This is the phase 2 data from the cohort of patients with obesity and type 2 diabetes. And you can see that the A1C control here is really profound and effective. And the weight loss in patients with diabetes is around 15% at the top doses here in this 36-week trial. So this is a really impressive weight loss effect in patients with diabetes that we know tend to almost uniformly, I think, lose a little less weight overall compared to patients with obesity and without diabetes. But fortunately, we have that data too with retatrutide, and the magnitude of weight loss here is approaching 24% at the top doses in a 1-year trial here. So this is, to my knowledge, the most efficacious benchmark for weight loss in a 1-year period with any anti-obesity medication outside of surgical interventions, I think. So the absolute weight change is really, really impressive. And I think this is going to have a huge impact when it comes forward. It's a once weekly again. And so at Lilly, within Lilly, tirzepatide once weekly, and this appears to be a little superior. It'll be interesting to see how they go on the market. I think innovation in incretin class is going to occur with small molecule chemistry approaches. I actually, I've got a slide here on orforglipron to introduce that. It's a non-peptide agonist. We call them internally an NPA, but it's a small molecule approach. Classically, that's an ugly small molecule, but it is a small molecule, and it interacts with human GLP-1 receptor only, so it doesn't work against the rodent receptor, and this is a fairly common against other small molecule GLP-1 receptor agonists. It's highly bioavailable and is appropriate for once daily dosing without a big food effect. So this has a lot of advantages. It's not the first GLP-1 receptor agonist. It's a small molecule class. That came from Pfizer with danuglipron, and that was, I have to congratulate them on their effort. It took 10 years. If you ever get the opportunity to hear the story of how danuglipron was discovered and developed, that is a really good story of brute force mentality and faith, and one of the good things that big pharma can do with big libraries and skilled med chemists. And now that this is a different chemical backbone, a different kind of chemotype structure to the danuglipron class. And now it's probably out in front a little bit of Pfizer's molecule. So we are hopeful. More data will be forthcoming this year in the ADA and EASD meetings. And I think we're all optimistic that this will be a really nice molecule. And what it does is elicit a large part of the biology of an injectable peptide. So this is the phase 2 data again from changing body weight in patients without diabetes. And you can see here that we're approaching 15% of the top doses. So it's clearly going to be a little less efficacious than an injectable GLP-1 receptor agonist, but it'll open up probably opportunities for a wide range of other patients to try a GLP-1 to see how it will work for them. And provide, other opportunities for combining different agents as well in an oral formulation. So we're very excited about this, and I think, now that a Class B GPCR has been cracked with a small molecule therapeutic approach, what other Class B GPCRs can be also similarly addressed and cracked using chemical approaches. So I think the example here with GLP-1 has triggered a lot of industry-wide effort and interest in other Class B GPCRs like the amylin receptors and so on and so forth. So I would expect more competition in the NPA space, the small molecule space, moving forward. So briefly mentioned amylin. I'm sure many of you are familiar with amylin, but it's also the poor stepchild in peptides relevant to obesity and body weight regulation. It's actually a pancreatic peptide. And is secreted from the pancreas in relation to insulin. It's a bona fide meal termination signal. So it actually really acts via the hindbrain through its receptors to shut off food intake in rodents and in people. The rat is the most translational animal model for amylin class of molecules and peptides. And it clearly induces significant weight loss through repeat administration or continuous infusion in obese rats, as well as in people. Amylin signaling is complex. It's a family of the calcitonin family of peptides, includes CGRP. There's a core 7-transmembrane GPCR called the calcitonin receptor, and it has a similar family member as well. But it requires an associated protein called a RAMP, and there are 3 versions of a RAMP protein that associate with the CTR to confer preferential signaling for either amylin or calcitonin like peptides. Really complex, I won't really go into it, but it's an important consideration when understanding how to translate from preclinical to clinical, and then which of the effects are driven by the different isoforms of the receptor. And so the key questions in this field are really, beyond just can it induce significant enough weight loss to be competitive, but do we want an amylin-like molecule or a calcitonin-like molecule, or both? Right? Most of sort of dual active agonists. And then how is the weight loss different from a GLP-1, right? And I think these are 2 questions that remain open and should, in my opinion, should be sources of lots of research. So the rationale for amylin comes from the old pramlintide for obesity studies that we did back at Amylin Pharmaceuticals. And there have been multiple, maybe 6 or 8 phase 2 studies for obesity that have been conducted with pramlintide. This was a twice-a-day injectable molecule, and not super easy to take, but consistently gave 8 to 10% weight loss in patients with obesity. This is an example of a BID regimen on the left. It's twice-a-day regimen on the left versus a 3 times a day regimen on the right. And the results are fairly consistent in terms of the magnitude of the weight loss that was observed here in these trials. This was also, you're able to combine it with things in the bad old days like sibutramine and phentermine and show additive benefit on weight as well as with GLP-1s, not in the clinical setting, but in preclinical models. And so amylin class of molecules is, in my opinion, going to be a space for innovation, but also probably the biggest weight loss therapeutic class outside of GLP-1s. And I think, at Lilly, we certainly talk about having a GLP-1 or an incretin portfolio and a non-incretin portfolio as addressing the majority of needs at this time that we can see. Of course, our neighbors here have invested a lot in amylin agonism over the years with cagrilintide. That's a sort of a classic dual amylin and calcitonin receptor agonist. I've just shown a little bit of data from their molecule here in combination with semaglutide, and these are a little bit of suboptimal dosing. I think they've probably got much better data than this. I could have picked a lot of different data to show, but I think this illustrates the concept that cagrilintide itself is a viable monotherapy option, and is also being looked at in combination here, and their combo is called CagriSema, and that's in late-stage clinical trials. They also have an oral single NME version that is a fused molecule that looks really quite interesting and quite potent in early clinical trials. So I think amylin and amylin GLP-1s will be an exciting innovation to go up against other just GLP-1 only type therapies. So that's kind of in the near term. Those molecules that I've just talked about are all in like phase 2, phase 3 kind of trials. Incretins will remain the backbone therapy with the amylin mimetic class in the near term. People, I work in discovery research, very early stage. So a lot of the things that we work on will come into the pipeline, hopefully in the next 3 to 5 years, and then in the clinical pipeline 5 years after that kind of thing. And I think it's, what do we work on? What are we looking to do to innovate in the obesity therapeutic space? And my bias is to say, well, we need more efficacy. And that's not a uniform opinion at Lilly, I'll tell you. And a lot of people will say, especially some of the clinicians will say, we're getting enough weight loss with retatrutide and tirzepatide. We don't need more efficacy. And I would argue that I think we still do. I think 25% or 20% weight loss on a 200 kilo person, they're still going to be probably clinically obese. And I think there's still metabolic benefit to be had. So it's that under response or the ideal response. I think there's still opportunities to understand why people plateau, why do they metabolically adapt to plateau for weight loss, and how do we alleviate that adaptation or extend the weight loss efficacy of our agents, probably through combinations, and that's going to require additional biologies and targets as well. We get a lot of quality of weight loss, and you would have heard a little bit about the loss of lean mass or muscle mass in response to the incretin class. I'll talk a little bit about how we think about that and what we kind of do to address that within Lilly. And then complementarity to the existing therapies. So it's what else can we combine that with and what other things? There's a multitude of other agents to enhance the patient experience, less frequent dosing, lots of other things that outside of just weight. And can we address other diseases concomitantly, like cardiovascular disease or kidney disease or liver disease with specialized combination approaches? So that's clearly other areas we talk about as well. Oral delivery, I've already mentioned. There could be opportunity of other classes there and longer acting therapies. And, I think people get a bit skeptical when I say we'd love to cure obesity because I work for a pharmaceutical company and, they're not really into cures. But I would say that's kind of the goal. We talk about how do we define and understand how to shift the body weight set point. It's, I would hope many in this room are working on similar kinds of questions. I think with the advent of really deep sequencing and molecular technologies, we hopefully will start to answer that question. Genetics in clinical trial participants will hopefully start to shape the architecture of what treatment response looks like. And ideally, yes, we would love to develop a therapy to get people to their ideal weight and take them off therapy, and they were able to maintain that with zero or minimal kind of interventions. And I think novel biology is definitely going to be required there. So let me talk a little bit about the body composition. I think this is the only slide I have on tirzepatide, and this is the change in weight from patients with obesity or who are overweight without diabetes. The SURMOUNT-1 data, this 22.5% maximum weight loss number is sort of the magic number until retatrutide came along. But it's not really about the weight. I think one of the things that came out from this study was seated back in the supplemental data, but it's the change in body composition. And on the left-hand side here is the total fat mass change, and on the right there, the total lean mass. And in the graph on the right, it's the ratio of total fat and lean mass, with tirzepatide versus placebo. The take-home message here is that weight loss is associated with reduction in both fat and lean mass. And I'm personally unaware of therapies that reduce body weight that do not have reduction in lean mass. We can argue about the magnitude and if that's meaningful or not, but I think it's in part a mechanical load sort of issue. But calorie restriction is associated with loss of lean mass too. No one ever really brought it up before, and it's clearly not impacting outcomes, as GLP-1s generally as a class are cardioprotective and have beneficial effects on other diseases. But is it a concern? And we don't know. We need more data. We need more body composition data from subjects in weight loss trials. But I think we also, as an example, just a thought experiment, can imagine that if we had a therapy that didn't have that, if we had a therapy that was able to specifically reduce fat mass without changing lean mass, then that might be more appropriate, or at least some patients might feel more comfortable. Imagine a patient with some frailty issues, or a patient who's older or potentially faced with a cancer battle, right? Maintenance of lean mass and weight there can be an important, marker for prognosis. So I think, if we had a choice, we should give patients the choice. But that's, so there's been a lot of work to think about that. And Lilly's made an investment in a company called Versanis that had an asset called bimagrumab. This was originally developed by David Glass and his team at Novartis, and this is an activin receptor inhibitor. Activins are a bunch of ligands that essentially regulate muscle mass and function, as well as adipose tissue mass and function. There are multiple selective and specific ligands, and they need two different types of receptors to work, the type 2 and type 1 receptors. So the type 2 receptors are ACTR2A and B, and bimagrumab is a high affinity antibody targeting both ACTR2A and ACTR2B. So it blocks the signaling of all the ligands in whatever tissue it can block ACTR2A and B in. And it really, the hypothesis here was that it would promote muscle mass, right? So you can get the muscle hypertrophy. It worked really well in animal models. Many of you would have seen the mouse and the pigs and the cattle that have mutations in myostatin, the same kind of concept. They have very muscular physiques. And in a clinical trial with bimagrumab, they saw reduction in fat mass. This is in a 48-week phase 2 study in patients with diabetes and obesity. But you saw that with DEXA, they could measure significant reductions in fat mass, in line with the reduction in body weight over the same time period with BIMA. And the key question here was, does it regulate fat mass and lean mass? And so fat mass is maintained there at the end after the cessation of treatment, and same with body weight. But what really happened with lean mass, and using DEXA for total or appendicular lean mass, or using MRI to measure paravertebral muscle mass, you can clearly see in the green line that there's a marked increase in lean mass and muscle mass in these patients over this time period. So we consider this to be a clinically validated pathway. The key question now is, the overall weight loss was nowhere near comparable to a GLP-1. What happens if you combine a GLP-1 with an activin blocker? Do you prevent the loss of lean mass in that context? So a guy called Paul Tichenor at Penn has published on this, and I'm just pinching a bit of his data here. So this is an animal study in obese mice. The graph on the left is a little bit faded, but you can see that in the groups that received semaglutide or the combination of semaglutide or bimagrumab, the weight loss is the same. So bima didn't necessarily add or enhance the weight loss here on top of semaglutide in the mice. It had very minimal effect on its own. If you look at the change in fat mass, despite no effect on body weight, you do see an enhanced loss of fat mass with a combination of GLP-1 and bimagrumab. And this is offset because of you only have two compartments to measure, but the lean mass is reduced still, but to a lesser extent in the combination than with the GLP-1 arm alone. So this is nice preclinical proof of concept that the hypothesis might be true. The phase 2 BELIEVE study is testing semaglutide and bimagrumab in people. I think those data will be available at the ADA in June timeframe, but it'll be exciting to see if this hypothesis pans out. And there's an ongoing study with tirzepatide as well. Okay, so just to the last few minutes, I will just talk a little bit about what we're doing internally in our lab and share a little bit of data. So part of identifying what you need to work on is trying to identify the patients that you want to help, right? So if we looked at the individual weight loss response from any kind of weight loss study, it's like a waterfall plot. We've all seen this, right? There's patients who don't lose much weight, maybe gain a little weight on the left-hand end of this curve. And there's the ones who lose a ton more. And then everyone reports the mean. And so what we're interested in is who are these under-responders? Why do they under-respond to tirzepatide or to semaglutide or to amylin? Are there any predictive factors for those that we could look at? And I was going to present some data, but I don't have it today. But the answer is no, not really. And are they resistant to all anti-obesity medications, right? So if you took those non-responders or under-responders and put them on to tirzepatide and put them on an amylin, would they respond at all? No one's done an experiment. So that's an open question. And are they the patients that we really want to target for future therapies? And then the other end of the spectrum is what can we learn from these hyper-responders? What makes them uniquely sensitive to GLP-1s? And what is the underlying genetic architecture or proteomic signaling that predicts treatment response? So these are the kind of questions we frame some of our research plans around. We do target ID. We do some internally, not classically great at it. Most pharma isn't. But I think we've set up some collaborations here, and I've just highlighted two. One is on human genetics with a company called 5 Prime Sciences, and the other is Fauna Bio, who leverage a transcriptomics-based approach in an animal model called a 13-line ground squirrel, which is a hibernation model. So they've taken samples over circannual timing periods and done a computational approach here, and also built in human genetics. So once we get some targets from our collaborators, and these are just two of some that we have, but what is the process we go to validate those? So you can do anything you want in cells, anything you want, but no one's going to believe it until you see an effect on food intake or weight in an animal model. So our goal was to try to shorten that time period from identifying which gene or target we want to work on to getting into the animal. So we've really set up an approach here to look for a guide RNA CRISPR-based strategy. And I see there's some posters on this too, which is really exciting because not much out there in the literature on how to do this in vivo in brain targets, right? So ideally, once we have our gene, we can validate guide RNAs in cells. We move into, package it up into AAVs, and we deliver it into the brain of animals. So when thinking about what kind of controls we want to use to validate the model, we immediately thought of leptin receptor. And so we did ICV delivery of an AAV carrying a short guide construct targeting the leptin receptor, and nothing happened. And we were a bit stumped, but we then redid it with intrahypothalamic injections, so bilateral injections to the arcuate nucleus of the AAV containing this guide, and we saw the expected effect of massive and immediate weight gain. This has been reported in literature before a few years ago, but it was really nice to see that we can recapitulate this. We've since done various flavors of this and combined different guides, trying to understand the fidelity of the model, and also a bunch of other targets to see if we can get some weight loss or prevention of weight gain to signal that part as well. And so I haven't got time to go through all that today, but one example that we kind of used it with early on is the gene bassoon. This was a target that came out last year from Giles Yeo's lab, and Rudy Leibel also had a similar publication at a similar time. Essentially, variations in bassoon were associated with severe obesity. And we thought, okay, great. So we should be able to knock this out in mice. And if it was involved in regulating body weight, the animals should gain a bunch of weight. So we went through the same kind of process, validated some guides, put them into an AV, injected them into the arcuate nucleus, and nothing happened. Right? So animals didn't move a bit. Okay, so how do we know we've done a good experiment? Right? And so we believe the in vitro data on guide, but how do we know it's getting in the brain and editing the gene in the brain? And again, with all the caveats that bassoon is widely expressed in the CNS, we don't know if this is the right region, all of that. But one of the ways we can do this is look at the indel efficiency in the hypothalamus. And so we were able to look at that through Sanger sequencing, and we're able to demonstrate that that was about 10% editing rate in the hypothalamic tissue from these animals, which is indicative, but it's not awesome. We're usually getting 60 plus, hopefully 80 or so in cells. And so we wondered if we weren't diluting the signal, right? If we're catching all the cells, but obviously the guides only get carried in by the virus. So it's really only the cells with the virus that should give you an indel. And so we're able to, this is not with the bassoon target, but with a different target. But we're able to dissociate the hypothalamic cells. We're able to flow sort on an M-cherry readout that gets carried in with the virus as well. And we're able to do Sanger sequencing on those neurons. And we're able to see, 85+% editing efficiency in the cells that got the virus in the brain of the animals. So we think we have a really interesting kind of workflow here and an opportunity to, never going to be high throughput, but hopefully somewhat rapidly evaluate multiple targets in the brains of animals for an obesity phenotype. So just to summarize, I think the take-home message here is that it's a really, really interesting and fun and nice time to be in obesity drug development and discovery. GLP-1s are the workhorse and are the big dogs in the room. Combinations, including with GLP-1s, will be the primary focus in the near and midterm. Small molecules will be the next wave as well. And the amylin class of therapeutics will be the next injectable, or the most proximal sort of incretin-independent or non-GLP-1 class of molecules. Activins are validated. Whether they can be viable therapeutics in combination remains to be seen, but I think the clinical data will come through soon. And I think longer term, the future, is can we use human genetics? Can we use deep sequencing and transcriptomics from animal models and specific cell types, to really find those tractable targets? Can we use markers to do that as well? And we have a pretty good internal capability at this time, but we are still growing in Boston, and looking forward to making an impact down the line. So thank you very much for your attention. Appreciate it.
Thank you, Jim, for a fantastic overview of current and emerging therapies for weight loss. It's really, as you mentioned, an exciting time to be in this space. So I'm sure there's a lot of questions, comments, etc. Anyone wants to open this up here to begin with?
Thank you so much for a great overview. I'm Birgitte, I'm from Novartis. Can you speak a bit more to the therapeutic benefits of not losing lean mass? Is that has to be seen as metabolic benefit, CV outcome, function? What you're thinking around that?
Yeah, I can't. I don't think that data exists. So I'm not aware of any data that suggests that maintenance of lean mass in the context of weight loss or negative energy balance is beneficial on any outcome. Will it need to be? Maybe. I think the way we think about it is not so much about maximizing or increasing lean mass. It's more about focusing on reduction of fat mass. I think there's a lot of discussion among some of the societies around obesity is more than just BMI, and you would have seen some of the position papers coming out around adiposity and percent fat is really where the emphasis should be at an individual patient level. So yeah, I think, as I said, I don't know. At the moment, if we're thinking about those therapies, we don't frame it necessarily as we get a clear outcome benefit. It's really around different kinds of biology and body mass or body composition effects, so people can have an option at some point, potentially, if they need to. Thanks for the question.
There's another question also down here, and there's up here. Let's start up here.
Yeah. Thank you very much for your talk.
Sure.
I have two questions. One may be less clever, so I'll start with it. Is the toxicity of small molecule could be a problem is it a problem in drug development with small molecules, that you make a small molecule and it's harder to predict the side effects? And the second question is, with drugs that affect appetite, do you see or do you expect to see also change in food intake preference for those people?
Yeah. Okay. So first question first. The answer is yes. Small molecules are much harder from a safety profiling point of view. Once you deliver them, it's hard to control where they go. The specific, theoretically, right? So everything gets tested in classic toxicology and safety pharmacology studies, but generally that's my experience, that they're harder to navigate that safety threshold. Doesn't mean we shouldn't do it, though, right? So some things are only druggable by small molecule. I think that second question on food preference, I don't know. I think there's some literature out there on GLP-1 shifting food preference from high fat to low carb, and similar for animal models and same for amylins. I think, overall, I don't worry too much about food preference. I think I worry more around the magnitude of inhibition and the durability of that response over time. Yeah.
We have a question up here.
Hi, thank you for the very nice talk. I'm just also wondering what your take on using these drugs, but in combination with lifestyle, how you envision this, because I thought that I might have heard, maybe correctly or incorrectly, please let me know, if you do resistance training, you can actually reduce the amount of lean mass lost. Is that something that you can reliably expect patients to do if you tell them, hey, please do resistance training, or can these drugs actually be better depending on different lifestyle factors, I guess?
Yeah. So I think the resistance training during negative energy balance, I think there's some emerging data around that. Hasn't been done at scale. I think people enrolling in clinical trials used to get a lot more diet and lifestyle intervention than they do now, but there's still some that comes with it. I think that you can tell people what to do, but if they do it or not, it's a wholly different thing, right? With bimagrumab, there's some emerging data too, that if you change to a more high protein diet, especially from the preclinical space, changing to a higher protein diet can also enhance the effect on muscle. So I think that there's a lot of learnings there. As always, don't take my word for it. I think, talk to your clinician, and it's a patient conversation to have with their doctor. Sounds like good advice to me, though.
We take the next question over here.
Yeah. Jose Flores from Mass General and Broad Institute, also from Boston. So
yeah.
A little bit of a provocative question, and I think you alluded to this. I'm not really sure what the incentive is for a drug company to cure obesity and reset the time point. As I figure, the only incentive is that you could price it as a one-time and done type of therapy.
There we go.
Which of course would be exorbitant.
Yeah.
And then would that then widen the health disparities, the global health disparities, because only some countries will be able to afford something like would be inordinately expensive.
Yeah. So, I think it's a good question. I like the provocative nature of the question. We don't generally come up with a business model before we come up with the therapy, right? So I think one hinges on the other. I mean, why not? I mean, I think, it's our mission to help patients, right? We really want to address patients, and cures are the best way to do that. Many people would even argue philosophically, should there be a cure for obesity, right? I think the chances of us finding the set point and adjusting that so you can tweak an individual weight to wherever they want it and then you're just done, I think are really, really, really small, right? But it doesn't mean we shouldn't strive to understand what are those mechanisms so that you can find additional therapies or different therapies that have better efficacy or better control or less intervention. And the long-term benefits to health should be apparent. Like I'm preaching to the choir, but if you reduce weight and obesity, so many follow-on benefits occur. So the value is probably there, even if it's if it's an exorbitant expense. But every therapy is going to get a little bit more expensive. Small molecules will become generic, right? In 15 years from now, 20 years from now, and they'll be the benchmark. They'll be standard of care, kind of like statins for cardiovascular disease trials, etc. But people are still having cardiovascular events. Heart failure is still a clear, prominent cause of death, right? So I think we have an opportunity. I don't worry about the business case. That's someone else's problem. I want to give them that problem. Yeah.
There's another question over here.
Yeah. Okay, thanks. Jens Halling, Novo Nordisk. Awesome presentation. So I agree with you that we're probably not close to the limit of efficacy yet, but we might be close to the limit of what is acceptable in terms of reduction in food intake. because of social consequences of reducing that too much. So that leaves us with energy expenditure for more efficacy. And I think you're probably the furthest ahead with retatrutide, which in increasing energy expenditure. But it also increases heart rate, and it's hard to imagine increasing energy expenditure much without, the heart having to deliver the oxygen, which might interact with the cardiovascular benefits. So what are your thoughts on that? And do you have any data on it, perhaps even?
Yeah, no. So, and firstly, apologies to all the Novo Nordisk folks in the crowd. I probably completely undersold Novo's contribution to this space, but that's okay. I think, so it's a good question. So yes, energy expenditure, expending calories through feudal energy cycles predominantly is kind of what we're interested in, purely as combination plays, right? So I think it's how you dial in that, how much of that you dial in, maybe even when you dial it in, right? So, but I think we would probably agree that if you prevent metabolic adaptation, you should extend that weight loss, that negative energy balance sort of line and plateau later at a lower point. The challenge is, understanding that. That may be a clinical question, but we still have to provide those agents in order to do that. We're not really that favored around, UCP1 based strategies, obviously, but glucagon, for example, I mean, we've asked internally if we have retatrutide, what is that going to look like in a cardiovascular outcomes trial, right? And, the expectation is that it would be similar or better than tirzepatide because of the additional weight loss profile. But when you start bringing in different modalities, different biologies, it's still an experiment. Phase 3 trials are still just experiments, right? You should be pretty, have faith that it's going to work. But I think at the end of the day, you can't anticipate all those outcomes. And it's more than just one trial, obviously, too, for different patient subsets and so forth. So yes, it's a consideration, and we try to map that out, and we try to get preclinical validation real early on about those kind of modalities. Yeah, hope that helps.
So another question up here.
Tune Pass, University of Copenhagen. A lot of the work and a lot of the targets have been mapped, and they act through the circumventricular organs for good reasons, and that's sort of where the focus has been and where a lot of focus is currently. What does it take, and how long will it take until we see targets that are not within, say, the media-basal hypothalamus and the dorsal vagal complex, but located in the PBN or amygdala or elsewhere in the brain, where you potentially have some biology that's impacting energy homeostasis?
Yeah, it's a great question, Tune. I think you're going to be one of the guys that helps with that. Like, I think, we have to, it's one thing to be able to identify a target in one of those deeper brain structures. It's another thing to get a molecule right there. Okay, and so it might depend on the type of molecule. Some small molecules have pretty good brain penetration. Maybe one way to harness it is to follow the neuronal pathways and signaling and try to get downstream or upstream of that to something that you can access better, which is, the literature better than me. There's much more emerging stuff now with the neurobiology of amylin and GLP-1 signaling, better tools available to map that and sequencing and transcriptomics. So it's coming. I think, we'll make advances.
Last two short, brief questions, relatively brief answers. Up here first.
Rishipan from University of Helsinki. So I have read about some of these previous drugs like semaglutide. They have some side effects like nausea and gastric issues and stuff like that. So what do you think about this activin or amylin? Do they have these kind of side effects or? Yeah.
So, I mean, the cagrilintide data suggests similar kind of major AEs around nausea, emesis, and GI, factors. Is it going to be exactly the same or a bit different? We don't know. We just have to let the data sort of play out. Bimagrumab, similar kinds of, injection site stuff. Something that's not GI maybe wouldn't have the emesis and nausea component, or at least less of it. Injectables is always weird stuff, though. You just can't predict it. Yeah.
Okay, final question.
Yes. Conchita, CBMR. So I wanted to know what your current standpoint is on the uses and misuses of obesity medication and the risks associated with this. That is, when people are taking obesity medication when they're not obese or have type 2 diabetes. Yeah. And just a final quick one. Can weight loss ever revert to a healthy metabolic state? And are we approaching it in our own way? Are we just putting a patch on something that will end up reverting back to that and healthy energy balance?
Okay, so first question, I'm going to completely dodge. That's a really loaded question to ask someone at a pharma company. Okay.
But you have to ask it though.
Yeah, yeah. I can't give you a good answer for that one around. Sort of, I mean, it's a good question. What about the off-label use of weight loss medications, right? And you know, it happens. Obviously, it happens. We don't do anything. We do a lot to avoid it, like as a company, as an organization. I think it's a good question for society and clinical practitioners. You know, they're the ones who gatekeep this stuff, right? So I think that's going to be a more stringent sort of evaluation of that need. You know, I think that weight loss is a very individual question, right? Where, you know, and there's both cosmetic, what I would call where the patient feels happier, like, you know, and then what are the medical benefits of that weight reduced state that they often can't see straight away, right? They might be able to say, oh, I'm getting off my blood pressure medications, or I can stop taking, you know, insulin or something if they're type 2. But at the end of the day, most of those benefits are sort of longer term on outcomes and things. And so really the weight change and the smaller pant size, that's the things that people see straight away. It's very clear if you stop taking semaglutide and tirzepatide, you put the weight back on a large part. So it's not a cure. If you want to maintain that weight reduced state, you have to stay on that. Will it be all that forever and always? Probably not, right? I think there is a lot of effort on understanding the weight reduced state in order to the physiology of the weight reduced state to understand if there's you know, interventions that we could have there that are much less frequent, like once a year type interventions that maintain that. But, you know, currently the weight reduced state is not a disease, right? At the agency, there's no regulatory path for this, but it's clearly a clinical benefit. It will just take a lot of time. People like Novo and Lilly being invested in answering that question and addressing that question to shape that landscape. So it'll take longer. But yeah, up until then, I think we'll probably have multiple kind of options, small molecule and injectable, that patients can try at different times during their journey.
Okay, with that, we will conclude the session. Thank you again, Jim, and thanks for a great discussion.
Thanks, Christopher. Oh, thank you.